So the first two lectures are really kind of covering a lot of the methods in cognitive neuroscience. So the first lecture last week, as well as dealing with some kind of conceptual issues, we went through what are called electrophysiological methods. And there were two of these that I went through. One is recording from a single, recording electrical activity of single neurons, which is done by implanting a tiny little thin electrode that records the activity around the actual neuron itself, which is obviously only done in a few circumstances. And then EEG, in which what you do is you don't go into the brain itself directly, you just place electrodes on the outside and you measure the electrical activity on the surface of the brain. It's obviously coming from somewhere in the brain. So with that, you don't know quite where in the brain it's coming from, but you can see how the electrical waveforms change, and you can correlate those changes in the electrical waveform with interesting things in cognition. So different aspects of face processing was what we went through last time. Both of these methods are what are called recording methods rather than simulation methods, in that you're just recording the normal activity of the brain. And brain imaging is also the same in that it's a recording method. That you can do those chairs for a second. It's a recording method rather than actual stimulation. Whereas neuropsychology, here these are kind of brain lesions. So here these are kind of naturally occurring brain damage. But also you can have, you can temporarily disrupt the brain by putting magnetic fields around the brain. And this is a stimulation method rather than a recording method. And we'll talk about this today as well. So with brain imaging, it's something that you'll have encountered, to some extent quite a lot in your lectures so far. What I really want to do is, though, kind of unpack this in more detail than you probably had before. What does it really mean? What is it that you're actually measuring when you put somebody in a large magnetic field and try and figure out what's in their head? And also, how can you use that information? What does it tell you about someone's mental state, about their what they're thinking at this moment in time? So most of brain imaging, so fMRI is the main technique, is used in the way that you give somebody a task and then you see what happens in their brain. Now the question is, can you also turn that on its head and just look at what's happening in somebody's brain and figure out what the task is? So figure out what it is that they're thinking, figure out what it is they're doing. So can you kind of turn the enterprise on its head and do what we could think of as brain reading? Can you just take the activity at any moment in time or over a series of time points to figure out you are thinking about this, you are doing that? Can we do that? So in order to do that, we need to think about what this is actually measuring and what the pitfalls are in kind of interpreting this data. And I'll come back to brain reading in the context of lie detection. Can you tell whether somebody's lying, telling a lie from just looking at their brain? So the basis of functional imaging is really what's is to do with blood flow. So fMRI and also PET, which isn't really used very much, are what are called hemodynamic methods. So hemo being the blood and dynamic changes over time. And the way that works in terms of how you go from single neurons kind of firing to actually measuring changes in blood flow is something like this. So when your neurons are firing, when they're generating action potentials, when synapses are active, they consume lots of energy, okay? And that energy is involved in pulling in oxygen and converting oxyhemoglobin into deoxyhemoglobin. So neurons firing uses lots of energy and it converts oxyhemoglobin into deoxyhemoglobin. And what happens here is that, that you then literally you dilate the blood vessels around those neurons in order to pump more blood into them, okay? So you've got two things happening. One is that you've got an initial kind of use of oxygen from the blood, and then you've got more blood being physically pumped into there. And this is essentially what it is that you're measuring these methods. You're measuring how the blood flow changes. But the blood flow change is linked to neural activity, and neural activity is the basis of cognition. So those are the kind of the conceptual steps in a nutshell. So the two measures here measure slightly different aspects of this process. So PET measures the blood flow in a region. So the way that it does that is, I'll come back to the slide in a minute. The way that it does that is that you actually inject somebody with radioactivity. What used to be done is a radioactive water isotope, which you literally have a kind of catheter in your arm, and the radioactivity goes into your brain. So imagine that you're using, say, your left temporal lobe to think. Then you will end up pumping more blood into your left temporal lobe, and it will show up as having more radioactivity in that part of your brain. Okay? This is literally how PET works, is that you just look which parts of the brain have more radioactive isotope. The radioactivity isn't in the neurons, remember, it's just in the blood flow that is feeding the neurons. Now, one of the problems with PET, aside from the fact that you don't really want a radioactive isotope in you, is the fact that it takes 30 seconds in order for the radioactivity to kind of peak and be measurable. And this is very slow in cognitive terms. So imagine you see a face, and it's going to take 30 seconds before you can measure that. So obviously the thoughts and the neural activity is instantaneous, but all these changes in terms of getting the radioactivity to the right place takes a slow amount of time. So this is another reason why PET isn't used so much. So the way that you would use it in practice is that obviously you wouldn't show just one face, you just show faces over 2 or 3 minutes, and that way you can do it. But you can't measure moment-to-moment changes in PET. So the method that's really taken over is this one, fMRI, functional magnetic resonance imaging. So fMRI uses a different signal. It doesn't use radioactivity. So this is one of its main benefits, in that it's seen as being kind of perfectly harmless really in most cases, well, in pretty much all cases. So the kind of the circumstances which you can't do it would be if you've got any metal in your body, which you think, oh, you haven't, and then all of a sudden you go to scan somebody and say, oh, well, what about my contraceptive cord? You think, no, you don't go in if you can't. Those are, you know, so things like, what are they called? Piercings can be a problem. It depends obviously where they are, but you don't want the piercing to kind of get sucked out by the magnet. So when you go into the room, for instance, if you kind of hold your keys like this, they would all kind of stand on end like this. And if you get closer, it would pull out of your hand. So any kind of metal in your body you would want to have. So provided you're metal free, it's seen as being perfectly safe. So pacemakers and things like that. So the way that this works is that you put your head in a giant magnetic field, in effect. So if you're not using radioactivity, what is the thing that you're actually measuring? The thing that you're measuring actually is the to do with the amount of oxyhemoglobin versus deoxyhemoglobin. So remember, as your neurons consume oxygen, you're converting oxy to deoxyhemoglobin. And deoxyhemoglobin kind of behaves a little bit like a magnet. It's got a slight kind of, it's called paramagnetism. It behaves in a certain way when you put it in a magnetic field. So you can measure the distortions in the magnetic field to figure out where you're kind of increasing your deoxyhemoglobin relative to your oxyhemoglobin. So you just use what's called a natural agent, a natural contrast agent. You don't need to inject something in order to see what's going on in the brain. Just this chemical process itself can be used with fMRI. So the thing that you actually measure is this thing here that's called the blood oxygen level dependent contrast. So whenever you see a paper that uses fMRI, you'll see this phrase bold. And all this means is that you're just using this kind of deoxyhemoglobin in the brain as the signal. It's called the bold response, basically. That's the thing that you're really measuring. And the way that this kind of bold response changes over time has what was called the hemodynamic response function. And this isn't to be confused with an ERP waveform. Okay? So this is basically what you are measuring when you're doing an fMRI experiment. Okay? And what you're doing is you're measuring it at each different point in space. So you're measuring this in the whole brain. And I'll come to that. So here, this is time along there. And this is your kind of, what's called the bold signal. It's the thing that you measure that's related to oxy-deoxyhemoglobin. And it has this characteristic shape. And the shape has nothing to do with electrical activity. It's all to do with the way that you kind of pump blood into that and the way that the composition of the blood changes. So why does it have this shape? So you can see it's got these three different things here. So what happens initially, so imagine that this is a part of the brain that responds when you show faces, and at time 0 you show a face to the person. So faces at time 0. What happens here is that the neurons respond to the face, they use lots of energy, and they convert oxyhemoglobin to deoxyhemoglobin, and that is this little bump here. It's called the initial dip. And in fact, the bold signal goes down because what you're doing is, if you will, this bit here is the deoxyhemoglobin, this is the oxyhemoglobin there. So the bold signal kind of measures the amount of oxyhemoglobin. So initially, you're consuming up oxygen and you get more deoxyhemoglobin, and this gives you this little dip. What then happens is that the brain says, you're using up lots of oxygen, let's pump more blood into that region. And this is effectively what you get here, that the curve shifts in direction and it's far bigger in magnitude. And this is actually pumping oxygen in there. So although you're using up oxygen, paradoxically you end up with more oxygen in the region than you would do otherwise, because you overcompensate by dilating your blood vessels and pumping more blood in. And essentially this is what you measure here. What then happens is that over time, you go from the arteries to the veins kind of relax, and the opposite happens, that you kind of flip over and then you reach baseline. So it looks like this. So imagine what you do is that you show the participants, say, another kind of visual stimulus, maybe a house. And this is how your bold response changes over time when you show it a house. Okay? And then what you do is that you effectively just look at the differences here. You kind of measure the peak or the area under this curve and do a t-test on it and say, is this thing here much bigger than that thing there? And if the answer is yes, you say that that part of the brain is responding significantly more to faces than to houses. And then what you do is that you, on the computer screen, you colour that part of the brain in according to how big your p-value is or your t-value and so on. So what you're actually seeing in these kind of fancy images of the brain is not actually neural activity. You're not even seeing blood flow, even though that's the thing you're measuring. What you're seeing is statistical significance. Okay? That is what you see in these images. You see statistical significance plotted graphically. And the way you get that is essentially like this. that you have multiple conditions and you do that. The reason why you need this condition here is that it always has to be something that's relative to something else. So all the time, your brain is using up oxygen. If it wasn't, you'd be having a stroke or you'd be dying or whatever. So it's always metabolically active, and some parts of the brain are more active than the other for whatever reasons. You've got lots of different kind of thresholds in the brain depending on how much oxygen they naturally consume. So you always need to have some other condition. You can't just put somebody in a scan and say what's going on, because the whole brain will be consuming oxygen, and some parts more than the others. So you need to always have this other thing. So that's one kind of natural limitation towards, you know, brain reading if you're just putting someone with a big magnet thinking out of a figure. There are also various other problems that stem from this. So in your statistics, you will know kind of the problem of doing multiple t-tests, okay? Is that if you say that you're willing to accept p is less than 0.05, your 1 in 20 thing, that if you do 20 t-tests, you might get something significant by chance, yeah? And it's very rare in a psychology experiment that you would have 20 different conditions all repeated 20 times. But that's effectively what you're doing here, in the sense that you've got your brain and you divide the whole brain up into little millimeter cubes. They're called voxels. It's like a pixel, but it's a volume unit. So you literally end up with tens of thousands of these voxels, and you apply, in effect, a t-test over every voxel. So what this means is that here you're running 20, 30,000 t-tests, if not more, and you will have, just by chance, 1,000 of brain regions being significant if you apply that normal kind of statistical threshold. So how do you deal with that and so on? This is one of the things I'll consider, but that's one of the problems, is that imaging is a statistical method, just like, you know, collecting data in your normal experiments, and you have to think about what the data actually means if you can get spurious results for that, these kinds of reasons. So any questions about this so far? So this is really what, you know, that's the nuts and bolts of what brain imaging is. Okay, so what does it mean when you read a paper on brain imaging and it says that it's active? Well, as I said before, it doesn't, you know, the brain constantly has a blood supply. So really all we mean when we say that a brain region is active is that it responds to one condition more than another. Okay, that's effectively what we're saying when we say it's active. It's a relative activation rather than absolute. This is active in this task more than that task, or when you see this stimulus more than that stimulus. That's kind of what it means. So it always involves selecting some kind of baseline or some kind of comparison condition. And really, you know, it's only active if it's greater with one than the other. So what this means is that whether or not you know, a brain region is meaningfully active depends on how well you choose your baseline, for instance. So if your baseline isn't very well chosen, then how you interpret that activity can be problematic. And I'll give you a concrete example of that. Right, so this is one of the earliest kind of functional imaging studies. It's not on pets, but it doesn't really matter for the story I'm telling you. It could just be, you know, to do with fMRI in terms of the way that it works. So what they're interested in doing is kind of mapping processes involved in essentially reading or visual word recognition in the brain. So they want to know what happens when you see the word cake, what happens in your brain in order to process that word. And really you could think about this involving various stages that we kind of know about from, you know, many decades of research. So you kind of imagine that when you see that, you've got mechanisms that detect curves, they detect junctions and so on, so visual analysis. And then you've got something that kind of maps that word onto a stored set of words. It's almost as if in your head you've got words that say, yeah, C-A-K-E, yeah, that's a real word, but P-A-K-E, that isn't. There's something that enables you to make that decision. So you've got like a store of the words in your head, and that's written word recognition, if you will. So you're not just looking at it in terms of curves and junctions. You're actually saying, yeah, this is something that is a recognizable pattern. Then there are other things you can do with it. You can get the meaning of the word, so you can generate, you know, its properties, that you could eat it, it's sweet, and so on. Or you can sound it out. You can effectively translate that word into its spoken form. You can produce the sound. So how would you go about studying where these processes are in the brain in terms of designing a good functional imaging experiment? So this is slightly complex, but I'll talk you through it bit by bit. So first of all, what you could do is that you always need to have a baseline relative to an experimental condition. So what Peterson et al. Did is that they compared, in one case, just viewing a plus sign versus viewing a written word. So what they're trying to do is figure out where in the brain this stage is here. Okay? So the idea is that when you see a written word, it involves visual processing, so detecting edges, junctions, and so on, but it also involves recognizing that as a familiar pattern. And the idea is that when you see something like a fixation cross, it involves visual processing, but you don't have to join things up into being a familiar pattern. Again, we can challenge that assumption in a minute. So the idea is that when so this one here involves visual processing, this involves visual processing and word recognition. So when you take that away from this, you're left with those parts of the brain that are involved in word recognition, but aren't involved in basic visual processing. That's the idea of this kind of assumption, is that you've got several processes involved and you look for tasks that have one thing minimally different. And you do get these kinds of regions. So then, then you would conclude that this is where written word forms are. But again, in order to make that conclusion, you have to assume that that is a good match to this. So well, what's the problem in having a comparing a fixation cross to a word? How do they differ?
Why wouldn't a string of symbols rather than...
Exactly, that's right. So yeah, so a better baseline would be having a string of symbols. So you could draw the wrong inference that this is involved in word recognition, but maybe it's just involved in looking at long objects rather than small objects, okay? So you would have made an incorrect inference about what that part of the brain does because you did not put, or did not think carefully about what your conditions are. So that would be one thing. So the kind of the gold standard in this field is not to use fixation cross. You have something that's called a false font. So you literally have something that looks like it could be writing but isn't. So people even use kind of other languages that people don't know. So you could show an English word versus a Hebrew word, provided you don't know Hebrew. It kind of looks like it could be a word, but it's not a familiar pattern to you. So that would be a better baseline for this than using a single plus sign, for instance. So here you've got, then they ask the question, which part of the brain is involved in seeing, in sounding out words? So here their baseline condition is something different. The idea here is that you just passively view a written word, okay? So you see the written word on the screen, and the idea is that that involves visual processing, involves word recognition. And in your experimental condition, you see a written word, but you say it aloud. Okay? So both conditions, you kind of match them visually, but in one condition you say it, and the other condition you don't. So the idea is that when you subtract from that, you get rid of visual processing, you get rid of kind of word recognition, this familiar pattern of letters, and what you're left with is effectively something involved in speech. Okay? And they get a whole set of regions with this. Again, there are various problems with this sort of baseline. So you might think that, well, what is that actually measuring? What it could be doing is actually the thing that makes your mouth move, for instance. Or is it involved in actually retrieving sounds? So here you might imagine that you do retrieve the sounds of the word, but you just don't produce the motor movements, okay? So how you interpret that, you could say, well, this is where the sounds of words are stored, but it might not be. It might just be this is where the motor commands to move your mouth are stored, okay? So again, you have to think what would be a better baseline for that here. Would it be saying it aloud in your head, or would it be, it's hard to imagine necessarily what it is. So the point is what part of the brain does kind of depends on how you kind of construct your cognitive models. And in a way, this is one of the reasons why people think, well, brain imaging is only ever as good as the cognitive model that you put in to test it. I don't think that that is quite right, but you can see where they're coming from. And then the last one here is what part of the brain is involved in retrieving the meaning of words, so understanding what a word means. So here they've got their baseline condition is then reading aloud a word. So you see the word cake and you say the word cake, for instance. So the claim here is that this involves visual processing of the word, it involves recognizing the word as a familiar pattern, and it involves phonology, articulation, producing speech, whatever you want to call it. The alternative one is that you see the word cake and you produce the verb that goes with it. So that most people would think of eat. So cake, you could throw, whatever, but eat would be the same. And the idea here is that that involves visual processing, word recognition. Here, both of these involve saying something aloud, right? They both involve moving your mouth and your lips and whatnot. So these ideally should cancel out, and what you're left with is kind of generating a semantic association or retrieving the meaning process there. And they report this region in the left frontal lobes that I'll come back to in a bit as being involved in retrieval. is that a good baseline? Well, it's not bad actually, but as we'll see, it's not kind of foolproof. There are other ways of doing this as well. Other ways of kind of designing your study that doesn't involve effectively subtracting one task from another. So this is another way of doing it. And all you're doing here, so what you've got on this axis is just the number of words per minute. So you're just listening to somebody saying aloud a stream of words. It might be just a list, it might be a sentence or whatever. I can tell you some more in a minute. And what you're measuring here is, it says RCBF on this, it's regional cerebral blood flow. You're measuring blood flow, it could be the bowl signal or whatever, it doesn't necessarily matter. But the point here is that what you're doing is that you've got 0 words a minute, which means you're not listening to anything, 10 words a minute, you're listening to slow speech, 30 words a minute, 50 words a minute. So here, you actually haven't got another baseline measure. All you're doing is it's almost as if you've just got one factor which has lots of different ways of varying. So you don't actually need to subtract something away for this because you've just got the different levels of the same variable. So it's a difference between having effectively a design that involves comparing two things like a t-test versus a correlational design, which is effectively what you've got here. And by having this kind of a design, you don't really need a baseline. It's almost as if each of the different levels of your factor act as baselines for each other. And you kind of, it's one way of thinking about it. And here you get these different kinds of profiles though. So this is the auditory cortex. So the faster your stream of input into that, the more your auditory cortex lights up, for want of a better way of doing it, of saying it. Here, this is somewhere that's near the auditory cortex. It's more involved in language. And this seems to show a rather different kind of profile. So what you have here is that it kind of flatlines a bit. So it responds to the presence of something that is language, but it doesn't care whether it's fast or whether it's slow. It just wants it to be almost present. So you've got this kind of angular shape where it kind of flattens, that is different. So you've got these different kinds of profiles. Here you've got an area, it's a different area of the frontal lobes, but it seems to be involved in working memory. And this shows a very curious kind of pattern in that it doesn't respond when speech is very slow, and it doesn't respond when speech is very fast, but it responds to somewhere kind of in the middle, I guess at a natural speech rate. But this is, again, it's interesting from a methodological point of view, because imagine that you've done an experiment that wants to compare how you kind of understand fast speech relative to slow speech. And you said you'd compare 80 words a minute versus, say, 10 words, whatever that is, 5 words a minute there. And then you say, then you kind of subtract one from the other. And if you did that, you would find that there was no difference in this part of the frontal lobe between fast speech and slow speech. And you would draw the inference that this part of the brain is not interested in the rate of speech. Okay? But that's not true. And you would only know it's true by sampling in the middle here. So again, you've effectively made I always get type 1 and type 2 mixed up. But anyway, you've effectively, this brain region probably is involved in, or is affected by rate of speech, but you're not detecting it, you've not put the right variables in. So again, the fact that you don't find it in the brain doesn't mean it isn't in there, you've just not entered the right bits. So the way you might think about this is that it kind of processes a certain rate of speech, and after a certain while it switches off. It's almost like when somebody talks so fast, you kind of, it becomes more and more demanding, and then it becomes, we can't process it, and then your working memory kind of effectively gives up, whereas your auditory cortex is always trying to kind of process. You've got these different kind of response profiles. So this is another way of kind of designing your here's a few other kinds of issues. So this is dealing with the problem of, it's what's called the signal-to-noise ratio, but it's also this idea that you're dealing with multiple t-tests, analysing each and every part of the brain separately. So what you've got to imagine here, that this little kind of grid are different kind of squares in the brain. Obviously, it's a 3D structure, so you're just looking at one slice through it. And what you effectively have here are each of the dots represents the bold signal, so the difference between them. And what you do with your fMRI data is that you do what's called smoothing. You smooth it, and you typically do this. You don't always do it. And this again sounds a little bit suspicious. Why are you doing that? And the idea here is that you're interested in big responses and not little ones. So there's a chance that you might get parts of the brain appearing to be active just because of the way you thresholded it. So for instance, you would make the claim here that maybe that part of the brain, this little millimetre here, isn't so important. So to smooth it, what you do is that you effectively add a normal distribution onto each of the little cells. So you kind of spread the activity of that brain region statistically to its neighbours. So what that would mean for a voxel such as this, which wasn't activated, is that it receives spreaded activation, smoothed activation from a lot of its neighbours, and it gets switched on quite well. Whereas this one here spreads activity out to its neighbours but doesn't receive any back. So in effect, it kind of dissipates it out. So you end up drawing a conclusion that this here is significant and that one there isn't. So it's almost kind of a winner takes all. But this also is taken into account in the statistics, is that you kind of threshold for the size of the activated area and so on. And this reduces the probability of getting little regions popping up by chance. Another way of doing it, obviously, is just kind of have lots of corrections. So if you run 20 t-tests, the idea is that you divide your threshold at 0.05 by 20. If you do that with brain imaging, you divide it by so much that nothing would ever come out. You would never see anything in the brain at all. So it would just be, if you divide your statistical threshold by tens of thousands of permutations, you just end up with nothing visible at all. So you have to make these kinds of compromises. And one of the things you learn to do when you do imaging is how to set thresholds, because it becomes a bit of smoke screens, different publications in different ways in order. So something can be made to be significant distance. So it involves kind of a good consensus as to how to do it. The other advantage of doing this is that it compensates for individual differences in anatomy. So imagine that the part of the brain involved in seeing faces is activated here in me, but in you it's activated there. But it's the same region, but it's just that your brain's a little bit bigger, or that part of your brain's a bit skewed. And the fact that they're there, if you kind of blur them out, it means that you end up with some kind of overlap that is meaningful. Whereas if you look at it at the microscope, you don't. Yeah.
I was just going to ask about the t-tests. So you compare each voxel to each other voxel, and then through time you compare them to each other.
That's right. Yes, you effectively have got various stages. So you do that comparison, and then you've got another stage that effectively looks for clustering of t-tests after that. So you do the smooth. Yeah, exactly. That's right.
So you have like a whole volume of voxels, and you look at each other, and then like 3 seconds later you've got another volume. Do you compare those two volumes with each other?
I see. Yes, no, you would average together all the different kind of scans, if you like. So, yes, so if you've got a volume of the brain in response to see one face, volume in response to see the other, you add all those different ones together, yeah. So when I report this here, that would be the sum of, say, 50 trials or 100 trials that you're comparing there, rather than a moment-to-moment thing. It's kind of the same problem with EEG, that any moment you've got, it could just be on and off for any reason, or fluctuating, and then this gets random. Yeah, so it's done at that kind of level. There are other kinds of issues with it as well, so I don't want to put you off with this exciting method, but it's not foolproof. So this is another kind of issue, is that we think that the bold response is linked to synaptic activity, as opposed to action potentials, by the way, which is the alternative. So here, you've got something that's excitatory, that this neuron is switching that neuron on. Here you've got something that's inhibitory, where this neuron is switching that neuron off. But both of these involve a loss of energy at the synapse, that it consumes oxygen, that generates the bold texture. So we can't tell from our bold signal from fMRI whether a region is being switched on or off. We can just tell that it's consuming oxygen and it is involved in some way, but it doesn't tell us which of these is true. And people have tried to figure out how that would be, and I think that the way forward is finding ways of imaging certain molecules, such as GABA, which is known to be inhibitory versus others, but that's not how imaging works at the moment. Most of the kind of synapses tend to be like this rather than like this. So some people say, oh, we always assume it's excitation, but you don't know. Right. So what I'd like to do for the second part is turn to what we call lesion deficit analysis. So these are looking at people who've had brain damage. So what you might find is that if you've got an adult who's had a stroke in their temporal lobes, that all of a sudden they might not be able to recognize faces, for instance. So this is something that's been around, since the 19th century, and it was a major, force in cognitive psychology for lots of years. And I think that there was a bit of an assumption when functional imaging was invented that old method would die out, and that functional imaging was the way forward. Okay? But I think that at least the more enlightened people see that that isn't the case. It's not the case that technology has taken over from this, but that the two methods are kind of complementary. And what I mean by that is that that in functional imaging, really the thing that you're manipulating is the behavior or somebody's task performance. You give somebody a task to do in the scanner. You ask them to read a word aloud. You ask them to generate a word, to just look at a word. And then the thing that you measure, your data, is the brain region, effectively, what is being switched on and off by that task. When you actually take a brain damage patient, the thing you kind of manipulate, in effect, is the brain region. You take somebody with a damage there, and the thing you measure is their behavior, is their ability to recognize faces, their ability to speak aloud, their ability to read, and so on. So it's almost if you're doing it the opposite way round. In one case, you're manipulating behavior and measuring the brain, and the other case, you're manipulating the brain and measuring behavior. So they're not, they're logically kind of almost the opposite. So it's not the case that this then becomes redundant. And what we would say here is that here there's kind of a stronger kind of causal link between the two. So we make an assumption that it is the brain lesion that causes that behavior. Whereas here, people say that imaging is kind of more a correlation, that you're looking what parts of the brain are correlated with what they're doing, but you don't know that it's necessarily absolutely vital for that function. And I'll give you an example of that. So this is a concrete example from the literature that I think is kind of quite interesting. And basically the question here is where is kind of conceptual knowledge, so semantic memory and knowledge of the meaning of words, where is this in the brain? And there's various kind of answers to that. So this is the part of the frontal lobes that's been implicated in lots of fMRI studies. So for instance, the study we talked about before, where somebody sees the word cake and they have to generate a semantic association, such as eat. You get activity in this part of the brain. Okay? But what's found is that if you ask a different question, if you ask the question, where in the brain does brain damage affect your ability to understand words, you don't get that region coming out. You get this region coming out in the temporal lobes. Okay? They're nearby, but they're not the same region. They're different regions. So here you're asking different questions. In one question, you're effectively giving somebody a task, and you show this is active. Another one, you're asking the question, what part of the brain gives rise to these kind of problems? So what I'll do is I'll show you a little video of somebody who has got damage to this region here, who's lost their... Now there are two ways of kind of thinking about this. And I won't go in for all these, but So one disagreement is that imaging data implies that a brain region is used in a given task, but the lesion data suggests that it's not essential. So that's one possibility. So it could be that, that in fact this region is important, but somehow the brain, the patients haven't, there isn't enough power to detect it. Another possibility is that the brain region that seems to be important is involved in some other general cognitive thing that isn't specific to the task. So one claim here is that this is really where your semantic knowledge is stored, but this is involved in kind of organizing information or is involved in retrieving it, but isn't the actual store of it. So it's kind of what's seen as being cognitive control or executive functions. So if you're given a task such as producing a word for cake, you have to kind of think through the various options and eliminate possibilities. So one suggestion is that what this is doing is not actually storing the meaning of words, it's involved in that process of selecting from amongst competitors. And there is some evidence for this, because what you find is that if you give people this kind of semantic task where there isn't an obvious verb to generate, well, so let me give you two examples. So if I say something like scissors and you generate a verb, it's very easy, you can say cut. If I say cat or chair, it's not at all obvious what the verb is that goes with that. And what you find here is that this part of the brain is really involved in generating non-automatic associations. So generating a verb for scissors, you don't need this part of the brain, so it just comes automatically. Whereas generating a verb for cats is like the word stroke, presumably, but it's something you have to kind of think through the options. And what you find is that although patients here don't have these kinds of loss of knowledge, if you give them that task, they are also impaired on doing it. So a patient with damage here doesn't have loss of knowledge, but if you ask them to generate a verb for, say, cat, they struggle with it, but they can generate a verb for scissors, where the answer is obvious. So it suggests that actually this is involved in semantic memory. It's not the core region of semantic memory that you might expect it to be, but as soon as you give the patients the right task, you find that in fact, yes, this is involved in semantic memory. It's just not a global deficit in semantic memory. And when you kind of explore the imaging data, you say, well, it is involved in semantic retrieval, but it's not involved in all semantic retrieval. When you can get an automatic answer, you don't need that bit of your frontal lobes. When you need to think through what the answer is, you do. So this is basically you know, what is going on through here. So you've got two different things going on. Well, the other kind of question here, sorry, I'm kicking the book back, is why this isn't activated by the imaging studies. And this is, again, a little bit of a puzzle. So why doesn't the imaging study do that? And again, here, one answer might be that the baseline effectively cancels it out. So it might be that even when you see a word, you generate its meaning, even if you're not asked to do so. You just can't help but do that. So that all your baselines involve semantic memory, and you can't switch off your conceptual knowledge when you see words. And that's always tends to be in the baseline tasks, for instance, here. There's another reason that's to do with fMRI, this is a hard part of the brain to detect. It's close to your sinuses, which are full of air, and it distorts the magnetic signal. And there are some parts of the brain where it's hard to get a good signal. This is actually one of them. And that's another reason why it's often missed in brain imaging, but you see it when you look at brain damaged patients. Okay, so I'll let you read through those yourself. What we'll do is we'll take a break for 10 minutes, and then we'll talk about lie detection. So how can you use brain imaging to figure out whether somebody's telling the truth? Okay. So lie detection. So the CIA, amongst others, and there are some private companies in America and so on who kind of invested a lot in lie detection, thinking that this is the way forward in the war on terror and all the rest, you know. But I think there's an interesting kind of science question here in terms of, you know, what we really can kind of get from the brain and so on. So the traditional kind of Jeremy Kyle style polygraph is not measuring anything to do with the brain. It's measuring something even further downstream. So it's measuring things like sweating, heart rate, breathing, and so on, for instance. which are seen as kind of being the emotional response to the lie rather than the lie itself. Because the brain is the organ that generates a lie, it is a seat of cognition and so on. So in principle, there is absolutely no reason why you couldn't study, get a lie from within the brain. It's a question of how you would do that and what the methods are. What's the appropriate baseline for a lie, for instance, to do that. So what I set is that this paper by Langelbaum et al., which has something called the guilty knowledge test. It involves playing cards. And what I'd like you to do is just think about the methods used here. If you didn't read it, then what I'd like you to do is just think about how you could use fMRI to do something like lie detection. What is an appropriate baseline? What are the kinds of issues to consider if you were designing, functional imaging study to study lying, irrespective of this? So I'll give you 5 minutes just to talk in small groups, and then we'll pull it together in a. But people do use this kind of brain reading approach, and it's interesting how it's done. One way in which it's done, actually, is using this sort of thing with what's called pattern recognition with unsmoothed data. So you can only do this in individual subjects, but you can do it in individual subjects repeatedly. And what you're effectively looking at is kind of patterns of activity in the voxel. So it's very complex maths that do that. But what you can find is that, for instance, you can see whether somebody is thinking of a cat versus a chair just by looking at the pattern over kind of millimeter scales in the brain, using that kind of method here. So you're not just looking whether region A is active versus region B, you're looking at the particular kind of pattern within each person's brain. And this is something that's been done, including with truth and so on. So, for instance, the brain response can be used to predict which object is seen or imagined from a limited set. So here they had 10 different categories, but then you choose on a binary thing. So basically, it's 90% accuracy with a baseline of 50%, but you've effectively shown, in order to do that, what you need to do is show the person lots of faces, lots of houses, lots of cats, see how the brain responds, and then you show them unknown stimulus X and try and figure out what unknown stimulus X is from seeing it. And the cool thing about it is you can also do it from imagining it. So it's a self-generated representation of a cat or a house. You can figure out with that level of accuracy what they're...
They featured that on the BBC Idea.
Did they?
Yeah, the Science Club.
Oh, cool, right.
Yeah, and they also did the lying fMRI.
Ah, would you put the link on the forum,
Yeah.
and then that would be great. Yeah.
There have also been studies where instead of reading what they're seeing, you actually predict the choice someone's going to make.
That's exactly right. Yes.
Before the person themselves knew what they were going to make as well.
Yes, exactly. So this is that they're given a choice of 2 tasks. I think one's like a maths task versus doing something else. And on any given occasion, they can decide whether to do the maths problem with this one. They can predict with the brain, again, relatively accurately. The other one that's been done is it's listening to speech, isn't it? That you play somebody, it's been done both with images and that you play somebody's sounds, and then you look at their auditory cortex and you can figure out what they're hearing, and you can almost play it back. And it sounds obviously like distorted speech, but that's just playing it back from measuring the pattern in the brain, if that makes sense. Again, it comes down to that you need to pre-test those. So you need to know in each person's brain how it responds to choosing between a maths problem versus whatever the other problem is. You can't just put something in there cold, and you need multiple trials. These are going to be basic kind of limitations for any of this kind of brain reading. You don't get it trial by trial. I don't think you ever do. Right, so yeah, these are the limits on brain reading. So each brain is subtly different anatomically. So imagine that you knew exactly in my brain where, when I see a cat, when I imagine this, when I do this, how the pattern of activity in the brain reproduces. You couldn't use that knowledge to transfer it to somebody else in order to figure out how they process cats. So their brains will be anatomically different, when you're dealing with things on the millimetre scale, if everything shifts by a millimetre, the whole model is completely wrong. You need ways of kind of adjusting it and so on. So although all our brains, you know, operate in grossly similar ways, when you come down to the fine level, it's not really the same. Also, you can measure kind of the general contents of thoughts, the extent to which you can know that you're thinking of a face or seeing a face and not an object, but you don't necessarily know whose face. Maybe it's been done actually, it's only a matter of time, but I'm not convinced you would get there to knowing the specific information. But there are interesting changes going on. It's also, you need to look at kind of conscious and unconscious. It's not easy to distinguish between the two as well, whether somebody is kind of unconsciously pressing or consciously pressing the brain, for instance, just from looking at it. So it might be that the brain's seeing it, the person's saying that they're not seeing it, and that could be the truth. It could be, you know, that really you can decode it from their visual cortex, but they're not seeing it because that information is not propagating or needs some other information or for it to be consciously visible. So again, here, that's also important for lying, is that the brain might know, but the person might not know, if you will, in the sense that they can't access all the information in the brain verbally, all the question. Okay, so the last 20-30 minutes is really talking about not brain imaging, it's talking about the opposite approach of, people call it reverse engineering. It's the idea that you, instead of giving somebody a task and looking what's happening in the brain, you actually manipulate a brain region and see how it affects the task. It's turning it on its head. So one of the classic ways of doing that was to disrupt brain function by naturally occurring damage. So you take a group of people, you damage their anterior temporal lobes and see what cognitive abilities they have, which cognitive abilities have lost. And this is called neuropsychology. The more modern way of doing that is to take a normal brain and just temporarily disrupt it by applying electromagnetic fields. So this is a la Penfield, you know, before, but using much safer technology. And this is called TMS, is one method, transcranial magnetic stimulation, and TDCS, which is it's direct current stimulation. But I'll talk you through that one slowly. So really what we're looking for in neuropsychology, so patient-based neuropsychology, is we try to figure out how cognition is structured and also how cognition maps onto the brain. So we can ask two questions here. What functions are disrupted by damage to a given region? So you can take a group of patients who have damage to a particular region, and you can ask the question, you know, what are they good at? What are they impaired at? Again, there are some problems with this, so you're never going to find patients who have exactly the same brain damage as each other. You know, they might differ in severity or other things that are affected. So, you know, people say maybe you should use single cases, but this is one way of linking, you know, brain structure to brain function that doesn't use function imaging. The other way of doing it is effectively not to ask the question where in the brain are they, but just to ask, you know, which functions can be selectively impaired whilst leaving others intact. So we encountered this last week in the idea that you can have language comprehension spared and language production intact, and the opposite one. This suggests that language can be, you know, fractionated at least two ways. It can be fractionated in many ways, it turns out, but these were the two kind of classic ideas. But again, you can make that inference just by having two patients with the opposite profile. You don't necessarily need to know where in the brain language production is. So you can use patients in different ways. One to kind of tease apart, you know, how cognition is structured, and another way to kind of link where cognition is in the brain from the brain regions. So, for instance, this is, you know, the classic idea from patients looking at the structure of cognition. So these are patients who have writing and spelling problems, for instance. And you've got one patient who, such as this patient here, who is very poor at writing and spelling vowels. So when they're asked to write down words such as Bologna, they miss out all the vowels. For instance, can write down the consonants. Very intriguing. Obviously, they could speak, so they could say vowels. It seems to be almost that they've lost the graphic representation of vowels. Maybe vowels are just harder, but it can't be that vowels are harder, because you can have patients with the opposite. So this would be this patient here, for instance, is a patient who makes very few errors on consonants and lots on vowels. But you can get exactly the opposite patient here, who makes lots of errors on consonants and not many errors on vowels. So it can't be that one is harder than the other. And this is basically the idea of double dissociation. And we conclude from this that within the cognitive system there are separate neural resources for consonants versus separate neural resources for vowels. And you can effectively do this for two patients. And you can also do it without knowing where in the brain the consonants and vowels are. You don't necessarily need to know that.
Is that the same when they're typing?
Yes, it's curious. Yeah, this is what they report. So this is CW, which one is CW? So this is a patient who makes lots of errors on vowels. So in this case, when they're missing, it's almost if they don't know, but if they're asked to guess, that they put the wrong ones in there. So they do get it from typing. So it suggests that it's not just at the level of the kind of moving the hand to make the strokes. It's something more abstract, yeah. So this is a patient here who has problems, errors with vowels. I think it's the second patient reported in this one here that they made from there. Again, it's very intriguing. I won't go into consonants and vowels, it's just the idea that you can go as specific as that, potentially, in these. So we use these double dissociation to infer that two tasks use separate neural resources, really. But the other way of doing that is to effectively group patients together to figure out where in the brain something belongs. So potentially you could take a group of patients who have deficits in consonants, those of vowels, and put them together. The problem is that if the brain damage is kind of like this, then you're not necessarily going to separate them out in the same way as, you know, with the others. It might be that you've got separate neural resources, but it might not be in regions like this. They could be in regions like that can be damaged. But as soon as you put all the patients together, it just looks like a mess. So you don't necessarily. It depends on the resolution of the imaging techniques and your ability to kind of map onto individual brains. So you've got the same problems when you're dealing with patients as you do with normal people, in the sense of making some of the mapping. So here you've got various ways of doing it. So one way of doing it is that you effectively take behaviour, so a collection of symptoms or a single symptom. So you take a group of patients who have one particular symptom, and then you find out where in the brain is there damage, and then you can kind of conclude that you know, that this might be relevant to that. An alternative way of doing it is that you take one particular brain lesion, so you take, say, a patient with damage to this region, you figure out what they can't do. So you've got these two different approaches. Either you find patients who have a certain behaviour, you find out what's wrong in the brain, or you take patients with damage to one region and you find out what's there. It sounds very straightforward, but you do get these kind of complex things. So if you do this, if you take, say, patients who exhibit a certain behaviour, and then you find out where the damage is in the brain, you might find that there are three different parts of the brain that can be damaged that give rise to, say, problems with writing letters or problems in reading. You might end up with three different ones. If you take this approach and you just take one part of the brain and see what it is, you might say, this part of the brain is involved in reading because when this is damaged, it goes down. But it doesn't tell you about the other two parts, if that makes any sense. Whereas doing it this way around kind of enables you to detect multiple parts of the brain that are involved with it. If you do it this way, you're in danger of saying that this is the part of the brain that does X, where in fact there could be other parts that do that. So again, you've got this interesting thing as to whether the thing you're measuring is the brain, or whether the brain is the thing that you're setting and the thing you're measuring is the behaviour. And the two are not the same thing. You can get quite different answers depending on what you feed into the other. And again, there isn't a right or a wrong necessarily. It just depends on what your question is. So in the example I gave before with generating verbs, people were interested in looking at where that, doing this approach here, because we knew from functional imaging that this region of the left frontal lobes was involved in tasks such as going from cake to eat. You see the word cake, you generate eat. We know that that was the case. We wanted to see whether or not it was really important. So you go to patients who have that. So you take a lesion location because this is what is driven by your hypothesis from the imaging. Whereas the alternative approach is to say, well, let's just look at patients who can't do this task and see what parts of the brain could be affected. Okay, so let's move on to TMS, because that's more interesting. We will come back to patients a lot, actually, in the next lecture. So TMS is basically, it comes under the umbrella of kind of disrupting cognitive function, but only very transiently. And what you're doing here is that you're applying the same logic as in patients, really, in the sense that you apply a brief magnetic field over a part of the brain and you see what the ensuing behavior is. So it's exactly the same as this third column here in terms of the logic, except it's not a real lesion, you're just temporarily disrupting the brain. Have I got the picture of it? Looks like, oh, I know this is a video.
I'm Roger Highfield. I'm the science editor of the Daily Telegraph. Now, words mean everything to me, but now I'm going to have the speech area of my brain turned off with a giant magnet. Humpty Dumpty sat on a wall. Humpty Dumpty had a great fall. All the king's horses and all the king's men couldn't put Humpty together again. Humpty Dumpty sat on a wall. Humpty Dumpty had a great fall. All the king's horses and all the king's men couldn't put Humpty together again.
That may have looked like a strange party trick, but it's actually part of a serious research project conducted by Prof. Vince Walsh in a basement lab at the Institute of Cognitive Neuroscience in London. He's using electrical and magnetic brain stimulation to understand the most complex known object in the universe and how to treat conditions such as migraine and stroke. Just to show the power of the magnetic fields passing through my scalp, and that this wasn't a trick, this is what it did to our videotape, even though it was positioned a few meters away.
Star Trek. Yeah, very left eyebrow twitch.
It's hard to see because of the interference, but here I could still sing a nursery rhyme, even when he disabled my language centre, because singing depends on the other side of the brain.
Okay. Humpty Dumpty sat on the wall. Humpty Dumpty had a great fall. All the king's horses and all the king's men couldn't put Humpty together again.
I felt a bit apprehensive during...
So it typically doesn't produce things that are very noticeable like that. So the effects can be quite subtle. You know, often it disrupts your reaction time by like 50, 100 milliseconds. You don't even notice it's having an effect on your brain. But again, the interesting thing there is how you use it. So the idea is that when you disrupt the language part of your brain, by putting it on the singing part of the brain, you can still produce the melody. It's just the words that kind of go, so the melody and the words can kind of be separated. But the interesting thing there is that it's also done in kind of real time. So one of the problems that people have, or one of the problems in studying brain damaged patients, is that their brains reorganize as a result of their damage. Here, what you're studying the brain function, it can't reorganize, it's just a temporary kind of disruption. Over milliseconds, really, or seconds. So you're looking at the real workings of the brain rather than the potentially reorganized workings. So here you see what's called a figure of 8 coil, this kind of shape here. And what that does is it's got wire going round it, and you have a rapidly changing current through the wire that generates a magnetic field, and the magnetic field passes over the skull and causes the neurons under the skull to effectively fire to generate action potentials. And what that means is that because the neurons are kind of firing in response to the magnet, they can't fire in response to the task, in response to what they should be doing. So your language neurons are being triggered effectively by the magnet instead of by the signal to speak, if you will. So you've got this competition between an outside source of activation versus the internal source of activation, which is cognition in effect with that. So in a way, it's kind of like trying to do two things at once. It's a bit like kind of dual task interference, where the brain is being used for two things. One, it's being stimulated externally, whilst it's trying to perform some kind of internal cognitive function. And we can talk about it as a virtual lesion, but it's not a lesion. So again, people get quite, worried about the idea of brain stimulation, but of course, brain stimulation is what you're doing now by coming here and listening to me, and so the fact that you're doing it from a magnetic field is obviously slightly different. I should say also that this is quite different from fMRI. So fMRI is obviously a magnetic phase, but in fMRI, although your head is placed in a magnetic field, it's the changes within that give rise to the stimulation. Is that right? Well, with TMS, it's the rapidly changing current that changes the magnetic field, and that this induces the change. The magnetic field in a scanner is actually large, but it's not rapidly changing. It's a radio signal that changes in the scanner. So we know the physics of this better than it's broadly right. But there are obviously some cases in which you don't do this. So if you have epilepsy, you're not allowed to do it. And also there are ethics in terms of how many pulses you can deliver to somebody. But also it's used as cures of depression. So if you've got parts of the brain that are kind of underactivated, this is a way of increasing the activity of them. So depression, for instance, or even in things like Parkinson's and so on, it's a way of kind of stimulating activity in brains that are kind of dampened down, if you will. So it's used in.
Does that mean that if you were to stimulate the amygdala, maybe you would generate more fear?
Well, potentially, yes. One of the things about it is that it's not clear how deep it goes. So the amygdala wouldn't be the best example because it's supposed to be only quite superficial, so it wouldn't go there. But in terms of the basic principle, yeah. So, for instance, it would. Yes, yeah, that's right. But it could. So you could do it over the motor cortex and have kind of twitching of the fingers and so on, or vision you can generate.
Wouldn't it prevent it rather than generate it, though? Because that's what it's doing with speech.
Well, yes. So the thing is that when you're actually doing a task, obviously it's interfering. But after you've done it, it kind of almost increases its resting activity afterwards. So after you've actually removed the pulse, it's kind of goes through a more kind of hyperactive region. And the idea is that then it kind of reestablishes connections with other things. So the long-term usage isn't clear with this, so, of having it. But the idea is when you're actually having the stimulation, it's kind of interfering, but that afterwards it can be facilitatory just by it's effectively boosting that region. So there are situations in which it can be used to facilitate as well as impair. They're supposed to be focal, so the actual stimulation site is only about 1 centimetre. The issue there is that once you've stimulated that region, it connects with others, so it kind of propagates throughout the brain. It's not clear how that does it. So although you can't stimulate the amygdala, if you stimulate a region connected to the amygdala, you will propagate to some extent to there. The other thing about it is that you can, each pulse is only a millisecond, so you can actually investigate exactly when that part of the brain is crucial. and you don't get good temporal resolution from fMRI. So in fMRI, if you think about the bold response, it takes 6 seconds before you get that thing to peak. So it's a slow response in fMRI. This is much faster. You can kind of fix the TMS pulse every second. So as well as kind of generating twitches of the fingers or whatever, you can also generate vision as well. So here this is, there's a part of the brain called V5, which is involved in seeing motion. We go through this in the next lecture. But anyway, it's on the outside surface of the brain, so you can stimulate it with TMS. And if you do that, you might see a flash of light that kind of moves. If you stimulate the back of your brain, which is where your main visual cortex is, you'll see a static little flash. If you stimulate it, so here you see a dot on your right side, so you're stimulating your left part of the visual cortex involved in seeing motion. You see dots that kind of fade, or bars that kind of do this. But also it can, depending on how the brain is wired, it can also be used to look at how stimulation can sometimes improve function. So there's a part of the brain involved in seeing motion called V5, and this can disrupt your ability to find a target that's moving. So what you've got to imagine here is that you're looking at a computer screen with these displays, and you've got to find the moving X. There it is. So you just press the button whenever you see the X move. Okay? And on some trials there won't be a moving X, so you don't just keep going like this. You have to discriminate. And what you find is that if you apply TMS on here, then it slows down your ability to detect the moving target. You get slower. You get the same thing if you're finding a static X, but you've got movement. So here, you've got to find that one here. But having something moving in the background, like TMS, also does that as well. Because here you're using movement to discriminate between something that's static and something that's... What's curious, though, is that here you've got to find a green X. Everything is static and nothing is moving at all. But applying your TMS over the movement area helps you to detect something by colour. And that's very curious. Why would that be? And the idea here is that different brain regions are kind of in competition with each other or sending each other noise. So it's almost as if that when you're looking for something based on colour, your movement system is kind of sending noise to your colour system and stopping it functioning to its full effectiveness. And by removing the visual system or getting rid of that that signal, what you're effectively improving your ability to find something based on colour. So again, it typically is seen as being interfering in the sense that it does that, but it can kind of facilitate. It can kind of almost release something, from it, depending on how the brain is wired. So again, this is another kind of use of TMS. Okay, and this is another example, and then we'll leave it at that. Okay, so this is, we can ask the question, what does the visual cortex of a blind person do? Again, it's a slightly ill-posed question because you could be people who are born blind or late blind and so on. What we do know about people who are blind is that their visual cortex isn't silent. It's used it's activated by touching things, so reading Braille, for instance, or listening to sound. So we know this from, say, PET or fMRI, that when a blind person is listening to sounds or that they're feeling things with their hands, they activate their visual cortex. What we don't know from PET or fMRI is whether that is really helping them, whether it's functional or not. So it could be that they're trying to use their visual cortex for seeing and touch, but they're not actually achieving that. It's just being activated because, because they're trying to use it. But is it actually contributing to their ability to use their other senses? And this is something that you can explore with TMS, because here you're asking a question, you know, if I disrupt the visual cortex, do I actually affect their behavior to discriminate braille or to discriminate certain sounds, okay? So then you know that it really is kind of causally involved. It's not just trying to process it in a way that's kind of correlational, I suppose. So TMS can be used to do that. So this is a study by Cohen et al., where the task here is to discriminate letters using your fingertips, if you will. And they've got blind people doing it either with Braille letters or Roman letters, so like an embossed letter. And because sighted people can't use Braille, they just use an embossed kind of letter A or letter E that they feel. And here they have 3 different types of TMS. One TMS is what's called sham, so they switch the coil on, but it's not resting on their head properly, for instance. That would be sham, so you still hear the clicks of the machine, and so, but it's not on. Then you've got 2 other conditions. One is that you stimulate the visual cortex, which is the one in purple. The mid-occipital region is your visual cortex. And the other is that you stimulate where your finger is, your sensory motor cortex. So you've got a motor cortex and your touch cortex are kind of next to each other. And this is your kind of sensory motor area, the thing that's involved in tactile exploration of the fingertips. And what you find here is that in the sighted people, when you apply TMS over their sensory motor, the kind of tactile system, if you will, it impairs their ability to detect embossed letters. Okay. In blind people, it's when you put it over their visual cortex, it affects their ability to detect embossed letters, even though they're just doing it with their fingertip. So this suggests again that in blind people, they are really using their visual cortex in order to make these kind of tactile discriminations. And it's not just the case that it's kind of epiphenomenal, it's just happening, but it's not really doing anything. It's, you know, it does seem to have, to some extent, even taken over the normal function that's going on here. So you can look at this kind of reorganisation and show that the reorganisation is really working or not. And they also report other things here, that when you stimulate their visual cortex, that the dots aren't in the right place, or that you know, other curious things. It's like the dots are missing is what they can report in Braille characters. That's very curious. And lastly, here, this is just an example of using TMS in this kind of study here, where you're having to find something that's on a screen. But the difference here is that they apply the TMS pulse at different times. And here, this is whether or not you get slowed. So this is the timing of the TMS pulse here. And what you find is that when you apply a TMS pulse at different times, it affects your ability to detect different targets, whether a target is absent or present. So TMS between 80 milliseconds and 120 milliseconds affects your ability to find something from an array of other visual things. And in another condition, it doesn't here. So it enables you to address questions of when a part of the brain is being activated. And you can't get this from fMRI, because fMRI works over different kind of time course.